I am really delighted to be here. Thanks, Dr. Yogeshwar Rao, for being an instrumental to be here. Uh, I mean, it's a very different story compared to academics and industry. We are very much satisfied of making a bond on what we wanted to make, because that makes exciting, and we see NMR, PE, KIA, TLC, and we publish maybe JAX or Angioente. That's something we talk about, impact factor, all those things. But when you go to the industry, the important thing is that time, cost, volume, energy, marketability, regulatory. There are several aspects: competition, business opportunity for sustained business, all those things. It's not only NMR. Anybody can make and anybody can take and show the data. But at the end of the day, the business means, making consistent business without problems. How do you do that? That's the first challenge. I will walk you through some of the slides what we had. The important thing all of you need to be happy is that we see that in next 7 years, the pharma business is going to go about threefold. It's something very interesting. All of you can have an opportunity to, live your dream. 7 years is a big time, and if someone has a very fundamentally strong idea, it's really possibility to do that. And how one can do and what are all the things, nitty-gritty in going through that process is also interesting. We will walk through that. And other thing is that there are already 3,000 pharma companies. So one needs to be very clear what you wanted to do. What is your knowledge? How it is going to be important? How are you going to be differentiated time to time? And what benefit you are going to create, which is going to be important. And the America and other countries, they are very happy to get cheaper products because they don't need to pollute themselves. You are there to supply, because we are also looking how to improve our economy. Are we improving our economy? Yes, in terms, but at what cost? That's also we need to keep very important, that what is the cost that you try to do business and create a problem to the not only the society here but the entire universe. That's the another biggest thing one needs to address. This is once again emphasizing, that there is really opportunity based on the November 2022 report. Okay, so from 2000 onwards, we try to understand how you can take your product. It started in 1990, but it picked up somewhere in 2000 to the regulated markets, because the money is much bigger there. That's very important. So you look at how many patents are becoming off patent, and what is the value of that product, and what we can do that. And one thing we had a very fantastic system that we have been trained, well trained in how to do things in terms of chemistry. There are a lot of things we could simply do overnight, which people used to think that we used to, do this thing for years and months, and we used to do things in maybe in 1/3 of their time. That's where our, expertise in terms of training, in terms of knowledge, which was really helpful to become the position that we say today, India is the pharmacy of the world. From where it comes, it's all people, chemists, scientists, all the people who came to the industry and made this thing to happen. What next to happen? We need to really understand whether every time, every year, you are going to look at how many patents are getting expired. Only thing we are doing at one point in time, it was an opportunity. It's also really thing that we need to learn what is the pharmaceutical means. It's not only looking at NMR. It's also looking at making of chemicals, making of pharmaceuticals. Pharmaceutical means it's not a commodity. You make a 1-ton material. If quality is not good, you take a 20% discount. It doesn't go like that. If the impurity is 0.1 and if it exceeds 0.11, it's out of the box. You cannot, issue that material out of your facilities. It needs to be reprocessed. And today, if you do anything, there is a CAPA. Why you did, what happened to your process, where things went wrong, why it went wrong, what validation you have done. All these things needs to be answered. It is not something simple. You need a fundamentally strong science in understanding each aspect. That's where your strength is. How you can think differently. What makes that thought process very different than others in terms of cost, in terms of inventory, in terms of speed, in terms of turnaround time. There are several things in the process which one needs to look into. It's not only that making things, but how making things can be much better. That's what industry would look into. Okay, so if you look into competitiveness, the first thing is that any process you generate, because there are 1,000 companies, why your process going to be sustainable for the next 10 years? You need to carefully think that, what is that process I want to use it, and how that process is going to sustain for next 10 years. Then only we can have a business, we can make some money, we can continue to grow the business. Otherwise, there is no point in making another process which doesn't have any significant difference. But other thing is that consistency of the product quality. I said if there is a batch failure, until you understand why it got failed, you cannot make a batch. Regulators don't come and see that if you are not solving, understanding the problem of whatever has happened, then there is no scope for you to take next batch. Let me forget about that batch, let me take another batch. What does that another batch mean? What is it you are going to do? If something fails, what is it you are understanding? How you are going to do corrective action? So if you don't know how you are going to prevent what has happened, and you don't know how to do a correction, that's the regulatory problem. At one point in time, regulators used to tell about 2 decades back, you do what you have written, you write what you have done. So be truthful. But today, it's not that you do what you have written and you write what you have done, but are you knowing your process? Do you know to a certainty that you can produce n number of batches seamlessly without any problem? That's the technology. That's what it is required. So that's where I think to some extent we have large understanding. But in terms of a transition from university to industry, there are several things. I mean, one needs to know that, because when we are doing PhD or masters and things, we do repeat certain experiments, what is there in the protocol, and then say we have solid product, we have measured melting point. So what? We have done all those things. But when it comes to industry, we say that I filter and shake it and filter. In manufacturing, the shaking and filtering is not possible. It's a huge reactor. How do you shake? It's nothing possible. How do you filter? What spatula you would use? There's nothing like that. Spatula, filter, shake. Now we have seen that Professor G.D. Yadav was telling the worst thing to use the RB flask, where you say that you swirl and filter. So these are the things you need to be very careful about. I mean, the transition is very important. The understanding is important. The environment is important. The scale is important. The requirements are important. The quality is important. So unless you have the transition understood, for yourself to become an entrepreneur, you need to be clear that what is the requirement. Is that I am meeting those requirements? Is that I am prepared to change to those requirements, environment. And then it's not only that we make a product, we say that we measure the melting point. There are pharmaceutical products, more often it's more than 300 molecular mass. It would have certain number of hydrogen bonds, hydrogen bond acceptors and donors. Then only it can biologically go and interact with proteins and cell membranes or whatever it is. You cannot say that I'll give paraffin and let it be biologically active. It's a paraffin, there is no affinity. If you want to create affinity, you need to have certain things taking and giving. Those taking and giving are the hydrogen bonds or salt bridges or whatever it is. And those also creates a problem, because it can also have its own interaction depending on the energy, depending on the solvent, depending on the time of crystallization. Because you want to make any pharmaceutical product in a handling way, that is handleable material is the solid material. But when you make a solid material, it can have a different polymorphs. And different polymorphs have different biological potency because it's mere solubility issues. So it can be sometimes toxic, it can be sometimes not soluble, sometimes several issues would be there. So these are all the problems one needs to know. The physicochemical attributes itself is a science. Unless we don't understand, we say that we have made a molecule, NMR fantastic, IR matches, these are all in solutions. Is it XRD is correct? Is it DSC is correct? Is it any molecular physical property is correct? Which are all things which one needs to know that. And the other things are the stability of the product. How do you know that the product which you have made, everything has an intended shelf life? If you don't know how long, at what conditions the product is going to be stable, then you cannot market it. When I say that my product is 2 years shelf life, to the specifications the day one you release should meet at the end of 24 months. There is no change of specification for day one. There is no other different specification for the 24 months. At the day one or at the end of 24 months, it has to meet the certain specified limits of your product specification. That's also important. And the inventory cost. Inventory means you can A to B and C, you can get 1 kg of C may require 10 kgs of A and 2 kgs of B, or you can manipulate in terms of what scientific rationale one would like to use it. So that makes a large inventory. If you produce 100 grams and use 10 liters of solvents, then you cannot be storing 10 liters of a solvent for 100 grams in factories and release every time. That's the biggest part one needs to know what kind of solvent you are using in lab. Sometimes we take for isolating 5 milligrams 100 ml of solvent and we say that we have made a product. Please be conscious. Everything has its own performance in terms of whatever way you look into. When you say that, why I want to use 100 ml of a solvent for isolating 10 milligrams, you need to ask that. It's a scientific reason. Is this 10 milligrams require 100 ml of solvent? No. Hardly you need maybe 5, 6 ml. And what is the purpose of using that? So that transition, the inventory cost, if I use so much, how much of, for 1 ton of a material I cannot have 50 tons of inventory in my godown. Transportation of that material, release of the material, charging of that material into the reactor. All these things is the difficulties which one needs to understand that. And from where I get this material? Is it I need to import? Is it available nearby? If it's not available nearby, please generate a technology and provide so that it can be a reliable supplier to you. That's an important thing in terms of competition. During the course of our understanding, many times, you have 1,000 scientists and you need to know how to measure their performance. When somebody says, I made a product, somebody says, this is RMC cost, this is the turnaround time, this is the productivity. One of the first principle one needs to know that in terms of innovator product, when he puts into the market, the API cost would be less than 1%. If it is 100 rupees is the drug product, it's usually 50 paisa is the API. It cannot be more than that. It is impossible to survive if you have 10%, 10 rupees. And you have to make for 50 paisa. And when you say that that is the raw material cost, then when you go to the plant, then the overheads would be another 30%. So 50 paisa to 75 paisa is acceptable. So when somebody comes and tells, many times people ask, what should be the material cost? I used to ask, at what price you buy 1 kg of sugar? How many carbons are there? How many hydrogens are there? How many oxygens are there? How many chiralities are there? They say 60 rupees. You get 6 carbons containing 12 hydrogens and 6 oxygen for 60 rupees. And why we say that 450 molecular weight material should be 10,000 rupees? Why can't we have a logical thought process? What should be the per carbon cost? And how do I look into a process of making much simpler than what somewhere else who would have made that? So you need to have a thinking process that if I can get petrol for 90 rupees, that's the price at the end you get it. If you can get a sugar for so much, you can get something else. And why we say that 400 molecular weight, 500 molecular weight for 1 lakh and 2 lakhs? One needs to think some of those things. So the atom economy should be 70% and more. If you don't have a process, you cannot exist. And total volume, it cannot be more than 3 to 5 times. If you have 1 kg material output, I mean, you should only use less than 5 liters. That's what the volumes. In terms of process duration, less than 8 hours, because there is a transition. Energy, ideally it should be 20 to 60 degrees. You don't want to cool, you don't want to heat, you don't want to do so many things. And the quality should be individual impurities less than 0.06. When you say limit is 0.1, if I make a material of 0.9%, 0.09%, if I give it to another lab to measure, he will produce with 0.11. It fails. Because the instrumental tolerance itself is a difference. If I say 0.09, it is passing in my lab, in other lab it will fail. It may become 0.08 also. But the tendency to fail is much more. That's where we have today problem with regulators. We don't understand my instrument when I say triplicate or whatever it is. What is my lamp? I mean, how many hours it walked? Its reproducibility of sensitivity. So when you go to the another fresh lab where the lamp lifetime is much shorter, then its performance in terms of sensitivity could be very different. What you got a broad peak of 0.09 can become 0.11. It fails then. So when you say that 0.1 impurity, you should be at least a 60% of your limit. Then only the material can sustain up to 2 years. When I say that there is no separate specification at the end of shelf life, means it has to meet whatever the specification you give it today. And if there is anything API cost more than 0.5%, you need to really think of, is this process worth taking into the manufacturing? How many people are working on that? Why this should cost more than 0.5? Unless you don't understand this light is, so yeah. So unless if it is less than 1%, it needs to be alert. If it reaches to 1 rupee, 100 rupees drug product, it needs to be an important alert. I cannot be continuing my manufacturing process. So otherwise, we will produce and make, file DMF, go and spend millions of rupees, and you will not make any money. That's very important thing one needs to look into. So if you look at it, is the actual metformin is available 500 milligram tablet, 10 tablets, 18.7 rupees. The actual manufacturing cost is 500 rupees. And if you look at the price, it's less than 10%. It's a commodity, it's a huge volume. So that's why less than 10% is survival. But if it is a material of, say for example, 1 lakh tablet, this may be going maybe millions and millions of tablets. And if it is 10%, it cannot be a business. And if you say that atorvastatin, it's less than 4%, because its volume is different. So you need to be very careful about what we wanted to make. It's not that you say that this is the drug product and I can make to that cost. No, it doesn't work like that. And when you use your science and technology, this is where it is. What I know about of making this molecule, what I know about the technology of this process, what I know about different ways of making of that. So when you say that, you need to have diverse technological capabilities in terms of chemical, in terms of biological, enzymatic, catalytic, everything. Can I use enzymatic process? Can I use catalytic process? Can I use any modified chemical process? Unless you have that spectrum of knowledge, it's very difficult to progress beyond. And what kind of engineering principles needs to be used for your process? Is it straight away can go into the manufacturing, or something can be simplified so that the manufacturing can be smoothened? So those things needs to be thing. And the high quality to be produced, and cost competitive, productivity, fast turnaround. We have seen all those things. And one needs to do anything you do for a sustainability, you need to have your own patent IP. And let's try to generate it. When I joined Sun Pharma first, we used to tell that we can file a patent. The management used to ask, okay, we will file a patent, what does it give? to the company? Do you know that we need to maintain a patent? That itself is a cost. And if your patent doesn't solve my, the company problem, filing a patent doesn't have any value, because you have to maintain that. So you have to see that what is that value which you have generated, the knowledge which you have generated, how it is going to help to the company, how it is going to help in the product. In terms of, it could be a very small understanding, but you need to have a lead on that, and you need to become a leader from that. Unless you don't emphasize that, you don't do that. I mean, you need to think about that. So this is the thing, I mean, when we say that, how do you understand in terms of your processes? Okay, if we look into, say, metformin, that's something very evolved process with a cost of 500 rupees. And if you say hydroxychloroquine, I have given molecular weight, number of carbons and other things. If you look at from this, say favipiravir is the lowest molecular weight and highest the cost. It's impossible to produce with good cost and to say that you are making a money. Maybe it was sold at 107 rupees per tablet. You could get a profit out of that, but to sustain in the market and to be environmental friendly, you cannot manufacture. As a scientist, as a responsible citizen, you cannot be manufacturing that kind of a product. Maybe you could have manufactured at one point in time because there is no alternative. But you need to find a solution, otherwise it's impossible to go through such a process. If you look at, I mean, in terms of how the process needs to be looked, if you say metformin at 500 rupees, my carbon equivalent cost is 46 rupees. Professor G.D. Yadav was telling that in pharmaceuticals, the E coefficient could be 50 times, because you are not having, like petroleum industry, it's only refinery process, my waste is 0.1%. You are using 5 steps, 3 steps, 5 chemicals, 70% yield, the volumes could be very different. So, but what is acceptable as of today, it could be 50 times is okay. So you have a carbon equivalent cost of 56, hydroxychloroquine 535 rupees. There is a lot of scope to improve. Why I am giving this example is that you need to understand per carbon. If I convert from market to what is per carbon value, do I see something value is very different? You yourself can understand there should be a scope. I should need to look into this. And if you see pregabalin 9.72, in terms of atorvastatin 16, and favipiravir 197, this is useless process. And the other thing is that whenever we do things, we don't understand in terms of sigma. What is my consistency? We always look, I mean, my purity should be 0.1, okay, less than 0.1. I have made 0.07, I have made 0.04, I have made 0.08. 3 batches, consistency batches, 0.04, 0.07, 0.08. How is it possible that you have a validated process? 0.04 to 0.08 is 100% difference. It's not a validated process. You have a process, but that process is not acceptable even though it is meeting your specifications. At any time it can fail, because your understanding of the process of producing an impurity at a level of 0.04 to 0.08, you have failed. You need to define why my product of this particular batch compared to that particular batch gave 0.08. You need to define that. To that extent, you need to have a certainty. When I do 10 grams, when I do 10 kg, when I go to 100 kg, that certainty you need to define in your process. These are the critical steps. This is the monitoring steps. This is where things can go wrong. This is what you can get. This is how I can monitor. If you don't have that certainty, please forget. We are all good in making things, but if you don't know these principles, then at the end of the day, we are not going to make anything, and our industry is not going to do, and our education is not going to help to the country to go anywhere. And if you see that the first process validation, everything is good. The sigma is 8.3 and 8.36. PV2, the middle value is going 2.7. It's still passing. When 2.7 is fantastic, but it is very likely 1 out of 10 batches is going to fail. This is the indication. And it's very likely if one batch fails, you are going to stop another 3 months production. So that means you have lost your business by more than 50%. You are making a product, it is meeting product, it is meeting specification. We become strongly arrogant telling that I have made a product, it is meeting, but this fellow is failing. Who is that fellow? And it is becoming difficult for that fellow to make you understand why it is failing, why we are concerned. The concern is, please be conscious that when I make something, I need to understand. You have to be very firm in telling with 2.7 itself, it can be stable for 5 years. You need to prove that data. How will you prove day one it is going to be stable for 7 years? It's impossible. You need to have certain skills to tell you that. I mean, so that's a important thing in terms of validation. You need to know what I'm making. When you say you are meeting specification, you need to know that what level you are meeting at what reproducibility characteristics. Okay. So to give some example is not to demonstrate chemistry. So there was an opportunity of citalopram. Everybody was telling that citalopram is important, it cannot be made certain things. But what we did is that it took about less than 2 months. There are hundreds of patents. But we thought, I mean, is it we want to follow a conventional way of doing things? What could be unconventional? Because conventional everybody thinks. Unconventional, it's rare. I mean, but still you need to make a difference. I mean, unconventional always doesn't give you something, a huge difference in terms of outcome. But you need to be unconventional if already 100 patents exist. So our process, we say that instead of 38 kgs, we could make 54 kgs. Instead of 99% purity, we could get 99.8%. Instead of 200 liters of solvent, we could use 100 liters. What does it mean? We are able to produce, instead of 1 batch, our process, they need to take 3 batches. So that means if my batch process is 1 day and their batch process is 3 days, the output requires for the same quantity is 3 times more. That is the turnaround time. For a given quantity of product, how much time you take? If I use 3 days, then manpower is required, your reactor is blocked, your chemicals are required, your analysis is required, your release is required. This takes another 40% of your cost. So that much you save. It's not only that you produce faster, not only that you are able to make more product, you are going to make better product, but it's also you're blocking with a slow process for making other products opportunity. The other thing is that there was a patent by Novartis that on imatinib, it's oncology product. They said you cannot make a drug product using alpha form because it's needle shaped, it doesn't have flow properties, it doesn't have hygroscopic property it has, so it's difficult to make a product and make. Then we thought, I mean, if what happened is that there is always a NCA -1 filing date. Nobody has filed because of this, because they had beta crystalline polymorph which has another 5-year shelf life, I mean the patent life. So then we thought, can we make an alpha form which is stable, which has a flow property, which is non-hygroscopic. That's the molecule is known. But the problem is, can I make a material with flow properties, a material with non-hygroscopic? That's the challenge you have. We could do that. I mean, it's not something extraordinary, but we could do that. The opportunity, you see that in 2015, it was 4.5 billions. And we got 6 months opportunity. And we got as much as what has been predicted in 6 months. In terms of your innovation, it's not something you had spent extraordinary money, but in terms of what you have in front of you, an opportunity, how you can react to that, how you can solve that problem is something one needs to look into. Okay. So what is that we are doing all these things? We have fantastic knowledge, we have fantastic training, we can think differently, we can do things differently, we can produce large quantity, we can supply everywhere. But what is the return? We supply more than 25% of all medicines in UK, 40% in US, but in terms of return, it's only 3.5%. We do large hard work, but we get peanuts at the end. But it's good we are getting, but is it this thing which we are looking for? Can we do something different? The amount of resources, the amount of knowledge, the amount of experience, the amount of these things we are putting, we could also spend something very differently, look for something. So this is one individual story. I mean, there was a person, Michael Rioda. He is the Gilead chairman. Before 1987, he was affected by dengue. There was no vaccine for viral, these things like dengue and things. He was a medical doctor. He doesn't have any experience of pharma business, nothing. But when he was looking that point in time, AIDS and other things, he was of the opinion, why can't we produce viral, antiviral medicines? Antiviral medicine. He is only doctor, he is like you and me, no background of pharma, no background of chemistry, no background of NCAs and discovery, drug discovery, nothing. But he had a pain of dengue experience, what dengue means. But he wanted also seeing that what is HIV means, how people become very untouchable. I mean, people used to run away. His objective is, why can't I make medicines for HIV? Why can't I make antivirals? You see, that's people when they don't want to look at a patient, he wants to think of making of that, finding a solution that. He himself is not an expert, no experience on that, but still he wants to do that. But if you see that in 1999, he had found out the ambisome. Ambisome is liposomal amphotericin, which we have used during COVID time for antifungal things. And he also produced Tamiflu, antiviral product, which he licensed to Roche in 9 years. A person thinks in 1987 and produces an innovation and licensed to the Roche, biggest company in the world. And one of the product, I mean, the best company in the world in terms of antivirals is Gilead. Nothing to beat, whether it is Merck, whether it is Pfizer, whether it is GSK. Even last year, there was a product from this company to treat very difficult to treat HIV patients. That's the conviction you need to have. That's the kind of thought process you need to have. You need to be very different. If you have a knowledge, if you have a spark, if you think that you can solve a problem, please think. You can always solve a problem. You need people to think together. But you need to have that spark to do something very different. That's very important. But he's an individual without any knowledge, understanding, but he had a passion of doing things, understanding things, making something very different. You see that, I mean, before 2013, everyone talks about Pfizer and Lipitor because that made 10 billion after 10 years. 10 billion, it's a mega blockbuster. Anything going beyond 1 billion is a blockbuster. And this product, atorvastatin, made 10 billion in 2010s. And everyone used to talk about we need to make atorvastatin because it's a 10 billion product. But when we went to the market, we had to give at 90% discount. That means if Pfizer used to tell the day before the patent expiry, they were selling $100. And then when we came, I mean, the generics came, they had to sell at $10. There is a 90% discount. That's the kind of a money you get when you go for a off-patented molecule. But they could get. But this company, Gilead, they makes hepatitis C viral drug. There was no product until then, 2013. In 1 year, they made more than 10 billion. In 1 year from the date of launch, where Pfizer took more than 10 years to become 10 billion with atorvastatin, but this company made 10 billion in 1 year. That's something phenomenal. BMS was following them, Merck was following them, but this company makes the passion that I wanted to be antiviral product. I wanted to be known as antiviral company. And this company, if you see today, from 2010 to 22, this number 25 in the pharmaceutical map of the world. They are not like some of these companies of 50 years, 60 years, 100 years. This company is like 30 years of pharma, of some Indian pharmaceuticals with single individual. We have companies like Alembic 100 years. We had Ranbaxy maybe more than 2.5 decades, 3 decades, more than 4 decades. But where is that our thinking? We want to produce large quantity, but we want looking for 3.5% of the 1.4 trillion business of the pharma. But that's what we are thinking. But we need to be very different. I mean, one big success, please give big applaud. I mean, this, not enough sound. You see that Bharat Biotech, he is also an individual. He came in 1990s telling that his mother was asking, please come back to India, you can be here. He was asking his mother what I can do here. I mean, if you are feeling that you cannot be fed, you have only half a foot stomach, we will make you happy feeding you. But he comes, and in 1999 he produces hepatitis B vaccine. And we see that in 2020, 2021 for vaccine, we could get all of us vaccinated against the COVID-19. This is a phenomenal success. An individual coming and making, vaccines produce at 10 rupees a dose. When you say that affordable medicine, this is also affordable. I mean, we can pay 10 rupees for a vaccination. I mean, the Pfizer sells for $10 for vaccination. That's how the government price. But this is 10 rupees. And all Covaxin, this thing we got all free of cost, government subsidized money. So this is something, I mean, an individual, if you have a thought process, you can do things, you can always do things. I mean, you can find avenues of doing things. This is something. The other thing, this is another individual. He was making, I mean, in the organon, the PD-1 biological. And that was acquired by Schering. And Schering told, any product in the phase 1 or developmental, we are not interested. We acquired organon to take phase 2 product to make our company bigger. So they said, if you have PD-1, please stop it. This fellow, the scientist is convinced PD-1 is something programmed cell death, is going to be the future of the drug discovery in oncology. Today, if you say that Merck, they have more than 150 programs in oncology based on only this molecule. He was stopped for 2 years. Then after 2 years, Schering was acquired by Merck. Merck did the same story. Please, anything below phase 2, don't do that. We are not interested. And this fellow was doing something, hiding something, getting very meager amount of funding and things. He was continued to do that. There was a publication in 2010 in BMS, very relevant to this, and they saw this, and we have this molecule with us. And they thought of licensing to someone for a few millions. They realized the potential. And if you see that, I mean, today, the 1/3 of the revenue comes from that one molecule which was supposed to go for few millions. And if you see consistently, 2020, 14.9 millions. And in 25, it is going to be 22 billions, one single product. So that's the kind of, he was a scientist. He was not knowing what to do, but he knows this can be a something fundamentally game-changing process in oncology. That's how one single product could give 22 billions and more. And if you see that 2009 to 2018, the number of the small molecules versus biologicals are changing. And of course, if you see that, I mean, the Humira, which is 5 billion, last year it sold for 20 billion. This is the only drug in the history sold year after year continuously for 20 years and sold together more than 200 billions. You see the opportunity, 200 billions for last 20 years. So you need to see how to do a business, where is the business, it's all business means your knowledge, unsolved problems, people who could not see beyond something, because most of the times when we see that this is something yellow, we go by perception, this is yellow, I know this. This is an object. Beyond your perception, there is something. The perception and object, they are very different. Your mind is different. Your mind can perceive to try to understand that object. When Vikram Sarabhai thought of making satellite to moon, the object he was using bicycle, he was using bullock cart, all those things. With that technology, nothing is going to change. But his thought process, his mind, I can still send rocket, which you and I can't think of that object. He could see beyond that yellow color, there is an object, there is a phenomena, there are certain things, we can deal with that thing. You know, that knowledge, that intelligence. Intelligence is beyond your, your mind. Because you need to apply the sense, your mind, and your intelligence. You can be, practicing that. When I see something, why is it this color? When I see something, why it has been produced? Why this time? So the understanding of the object could be in various ways, with various phenomena, with various minds. It could be very different, and outcomes could be very different. So that's where you need to be very careful about when you say that, do I stop thinking here? If I like, where I can stop thinking? Because you can never dreaming for 100 years. There is a lifetime. If you have to dream for 1 month, dream it. But you have to be the best in terms of dreaming and coming out with the solution. And if you see that, the Pfizer, in 2020, they have started this small molecule dosing the pill. They had a vaccine from the BioNTech, but they also realized that a molecule with a pill could also be nirmatrelvir, could be produced, because in 2003 they had an experience in the COVID-like kind of a situation. So they had some kind of a background molecules and tried to do that. And if you see that, they could quickly assemble in 10 months. March 2020 they started, and 2021, December 21, they got approval. So you see advantage. We all think discovery takes 12 years. Discovery requires 1.2 billion. To become blockbuster, you need to be another 10 years. No. Situation. Whether we wouldn't have done India, yes, it's very much possible. But we don't want to venture that. How much this product sold last year, 10 months? The 10 months, this product sold more than 17.1 billion. This is the volume. In 1.5 years research, they could get 17.1 billion. This is the opportunity. So we need to see that where is the opportunity, how I can make it. Yeah, okay. So this is the kind of molecules, people are talking next year in terms of what it can bring, this thing. And this is another product, tirzepatide, which is being launched last year. They expect to be 25 billions in terms of the market, peak market sales. Okay. China, number of molecules IND last year 2020, 393. Where we are nowhere. No? And how many products in terms of phase 2, phase 3 clinical trials in China? Is that we are not a scientist comparable to that of China? We are nowhere. They are competing with China. If you think of next 10 years, all medicines, what they require for their population will be produced by China. They don't need any medicine to be imported or to be purchased from anybody. We don't have that capability. We don't have any medicine developed in India to be produced and to be given our own people. So what we thought last 4 years, high impact innovation, sustainable. Anything you do, it needs to be sustained, otherwise there is no business. You need to identify unmet medical needs, unmet problems, all those things. And we competed with one of the programs. There were 10 major companies. We could solve a problem, and we licensed to Sun Pharma for 125 million, this thing. And we have another program which we are competing with this company, tirzepatide. And we have at a very different profile compared to what has been produced. And we reasonably sure that it could be definitely much more competitive to what has been produced with these things in terms of various things. This is the program where we started, and we could complete in 30 months, and we are into phase 1 clinical program. So how do I identify? You need to identify scientific challenges, matching with individual perspective, unsolved problems, estimate market potential, define attributes and problems, create strategies. There are several things you can think of finding a solutions. So the Moderna co-founder, he was there in 2021. When you talk of new things, many people, they don't understand what you are talking. They criticize heavily. You don't need to be worried. That's a part of your game. If you are convinced about things, please go ahead, unless it takes 10 years. But if you are sure that you have a real problem to solve and there is a solution, you need to venture into that problem and you will succeed like the people we have seen. So you need to be really dreaming every minute of your life. If you think of after 10, 20 years that I could have thought of this problem. Yeah, you could have thought, but what you have done. So please react today. Otherwise, you would be wasting your time. The important prerequisite is you need to be creative and innovative, doing right things and doing things right. It's very important. You need to think right things and you have to think right. So if you don't think right, you don't do right things, it's also failure of things. Thank you.